Let us now showcase the variety of RIKEN's research facilities. We will present the characteristics of each facility, activities related to sustainability, and opportunities for European and other stakeholders outside Japan to build new partnerships through such facilities. The first on the stage is the RIKEN BioResource Research Center, which celebrated 20 years anniversary last year. Director Toshihiko Shiroishi will tell us more. So, Dr. Shiroishi, please.
Thank you very much for introductions. I'm very happy to introduce the RIKEN BRC for European research community. Can I share the slide by myself here? Thank you very much. Okay, so the RIKEN BioResource Research Center, abbreviated by BRC, was founded 2001. So this year we celebrate 20th anniversaries. So this figure is a key visual used in the many events of anniversary celebrations. This is the Mount Tsukuba in Ibaraki Prefecture, very close to the RIKEN. So the many bioresources were provided RIKEN, and it's circulated all over the world. So it symbolized the activity of RIKEN BRCs. The left and right phrases stand for our sincere gratitude to research community, and right side is our aspirations for future directions of RIKEN BRCs. Okay. So next slide just shows the mission of RIKEN BRC. It is quite simple. Research community produce bioresources, and we receive depositions or donations and distribute the bioresources with high quality and useful information. And the research community again develop the new resources by using BRC's resources, and we receive again and distribute. So bioresources are circulated between research community, academia, and industry, and RIKEN BRCs. So our mission is quite simple. We drive bioresource circulations and contribute to life science, innovations, and ultimately SDGs. To do so, we have 4 activities. One is collection and preservation of bioresources, mostly developed in Japan. And the second, rigorous quality control under ISO 9001. Actually, this is the international quality control management systems. And the integration and the standardization and the dissemination of bioresource-related information. That information is getting important these days. And finally, we conduct research and development to develop new resources and to enhance resource usability. In RIKEN BRC, we operate 3 programs. One is Bioresource Infrastructure Program. This is a core program. It's collect and preserve and provide bioresources with high quality and usability, high usabilities and informations. And we handle 5 resources. One is experimental animal, namely mouse, and experimental plants, mostly Arabidopsis and cell lines, including iPS cell line, human and mice, and genetic materials, DNA clones, and microorganisms. We also handle the important issues, integrated bioresource informations. So these are key programs. And the second is key technology development programs. It's aimed to develop novel technologies for effective and efficient preservations and quality control of bioresources. We focus on the mouse resources. And the third is Bioresource Frontier Programs. It's aimed to conduct bioresource-related research and development to solve social and urgent problems that we are facing. There are 6 teams shown here. They engaged in this third program, Bioresource Frontier Program. It's a research program. Here, I'd like to introduce the national project named National Bioresource Project, abbreviated NBRP. The Japanese government launched the NBRP in 2002. It aimed to strategically collect, preserve, and provide bioresources essential for life science. And this NBRP has been revised every 5 years. So the project in the last fiscal year, in the 4th terms, at present, 31 bioresources, which was shown here, bioresources, core facilities, and the information center are now selected for NBRP. RIKEN BRC joined this project. 5 resources, mouse, Arabidopsis, general microorganisms, human and animal cell lines, genetic materials are selected as a core facility of the 4th term NBRP. So BRC play a very important part of this national project. One unique feature of the bioresources in RIKEN BRC is that we collected cutting-edge bioresources which are deposited by Japanese excellent scientists. We are very lucky because these are 4 scientists, outstanding scientists. They are all Nobel laureate. Dr. Yamanaka, Dr. Omura, Dr. Osumi, and Dr. Honjo. They deposited almost all their bioresources to RIKEN BRC. Notably, they deposited their bioresources far before they received Nobel Prize. So this indicated that the RIKEN BRC was trustable for these excellent scientists. Their bioresources are widely used worldwide, and we can see the very good outcome from the users' publications, scientific paper publication, and also patent. So this is a time course of the expansion of resource repositories since foundation 2001. We have 5 resources, mouse and experimental plant, cell line and iPS cell line, including iPS cell lines, and gene materials and microorganisms. All these resources were expanded year by year. At present, the size or volume of the repository is in the rank of the top 3 worldwide, world and public bioresource center. So we are very proud of it. Then I'd like to show you our achievement of our distributions. To date, we distributed 291,000 items to all over the world in the last 20 years since our foundations. The largest user category is domestic universities, and second is the overseas academic institute and the universities. The outbreak of these categories was summarized here. Domestic 78%, for overseas 21%, for academia 84%, for profit 50%. So we contribute both to domestic overseas and academia and industry. The bottom 2 map is a geological distribution of RIKEN BRC users. For domestics, more than 7,500 institutes have used RIKEN BRC's bioresources. Importantly, there is no duplicate in the institute. For the overseas, over 6,000 institutes have been used bioresources from RIKEN BRCs. So both domestic and overseas, so many institutes use bioresources from RIKEN BRCs. And so how about the outcomes? I would like to just show you the contribution to publication of scientific papers and patent. Top table is the publication of scientific papers in the last 5 years. each year, very close to 2,000 scientific papers have been published in each year using these five bioresources from RIKEN BRCs. So cumulative publication is now reached to 27,000. And the bottom is patent publications, and each year 5 or 4 and 300 of patents have been published using these bioresources. And the cumulative patent publication since 2001 is now reached to 8,000. So the data clearly shows that the bioresources, RIKEN BRC contributed not only academia, but also the innovations, in other words, the industry. So following this slide, I would like to focus on contribution of RIKEN BRC to life science in Europe. To date, we distributed 26,000 bioresources to European countries, European researchers, to over 2,000 institutes in Europe. Top user is Germany and France, UK, and Spain, and Switzerland followed. This is an outcome from European researchers using bioresources from RIKEN BRC. Scientific paper and patent publications. The European researcher published 1,600 papers using RIKEN BRC's bioresources. And they published 120 patents using RIKEN BRC's BRC. So it's a huge number. In Basel, I'd like to show you here the contribution of researchers or institute in Europe to RIKEN BRC. So this table indicates the resource deposition from Europe to RIKEN BRCs. 2,288 bioresources have been deposited to RIKEN BRCs from the 139 institutes. So the top contributor is Germany, and followed by France, Sweden, UK, and Belgium. So these three data clearly demonstrate that RIKEN BRC has a very good relationship with European research community. I would say BRC and Europe are interdependent and mutually beneficial. We would like to keep this cooperation with European communities. I would like to touch briefly on the global network of bioresource center or related consortiums. Variety and quantity of bioresource produced have been increasing dramatically and already exceeded the capacity of single resource centers. And another issue is global standardization or harmonization of quality of bioresources needed. So we need global network of bioresource centers and related consortium. RIKEN BRC is now actively participating in such networks or consortium. I introduced one consortium, that is International Mouse Phenotype Consortium, IMPC. This consortium aims to generate catalog of function of all genes on mouse whole genome by standardized phenotyping of mouse mutants. BRC joined this consortium as a representative of Japanese research communities. After my talk, Dr. Jan Herald may introduce more detail of this consortiums. So this is our last slide, the summary of RIKEN BRCs. Specification of our activities is shown here, and user from Europe, and access to bioresources of RIKEN BRC, and how to use bioresources summarized here. And these are useful links. We are very happy if many European researchers use our resources and make a good outcome using the resources. We are very much welcome if the European researcher deposited their important bioresources to RIKEN BRC. RIKEN BRC has produced video movies introducing activity of RIKEN BRC as a part of its 20th anniversary celebration events. After my talk, I would like to show you two of them. One for introduction of whole RIKEN BRC, another for introduction of experimental animal divisions, which is in charge of mouse resource infrastructure program. We hope you enjoy these two movies. Okay, I stop here, and thank you very much for your attentions.
生命は神秘と驚きに満ち溢れている。私たちはバイオリソースの可能性を開き続けてきた。バイオリソースは生命が育む未来を開く鍵だ。私たちは科学が生み出すバイオリソースを集め、整え、保ち続ける。生命の謎に挑み、人類の未来を切り開く人々のもとへと届け、絶えず勇気を与え続ける。生命は無数の小さな宇宙の運動だ。私たちは一つ一つの宇宙の神秘に挑み続ける。私たちは共に知識と技術を磨き続ける。明るく豊かな未来への架け橋となるバイオリソースを通して、新たな真実に出会う驚きをあなたと分かち合うために、一人一人の命が守られる持続可能な社会を実現するために。私たちは科学が正確に進歩するためのインフラを整備しながら、バイオリソースの新たな可能性を発見し、今よりも豊かな未来を描く研究センターであり続けます。Foundation for Discoveries 理化学研究所バイオリソース研究センター。
マウスは人のモデル動物として世界中のライフサイエンス研究で使われています。日本でも感染症、免疫、がん、発生再生や脳科学の研究で遺伝子の働きを解明するため、さまざまなモデルマウスがゲノム編集や遺伝子組み換えにより搾出されています。私たちはこれら我が国で開発されたマウス系統を収集、保存、品質管理して世界の研究者に提供しています。こうした活動に必要な技術開発や新たな社会ニーズ、研究ニーズに応えるマウス系統を開発するのも重要な使命です。
実験動物開発室ではさまざまな系統のマウスを収集、保存、提供しています。スタッフの皆さんは普段どのようなお仕事をしているのか、ちょっと覗いてみましょう。
こちらは提供を受けたマウスの健康チェックと検査を行う解析研究棟です。世界中の研究機関から送られてきたマウスの健康状態を確認し、いろいろな検査をするところです。マウスにも感染症があり、ここで検査します。特に人にうつる病気には目を光らせています。病気にかかったマウスを研究に使うと正しい結果が得られません。繁殖にも影響します。実験動物開発室では健康で元気なマウスを研究施設に提供できるよう、開発室のすべてのマウスの健康検査を行っています。
複数の工程を経て健康状態の整ったマウスが保存されているのがこちらのバイオリソース棟飼育室。室内を正常な状態に保つために入室の際には スタッフも念入りに準備をします。こちらが飼育室の中。
正常度の高い部屋でマウスを飼育しています。同じ系統を同じラベルの色にして区別しています。それぞれのラベルにはマウスの系統の名前、マウスの生まれた日、さらにその子供の生まれた日が記録されています。こちらでは日々マウスの健康状態を確認したり、マウスのケージを交換したり、子供が生まれたらその記録をして、子供が大きくなったらまた新しいケージに移しています。マウスを増やしてそれを必要としている研究者の方にお渡ししています。間違えないように気をつけながら仕事をしています。
マウスの環境を清潔に保つための清掃作業も欠かせません。こちらは自動化されたケージの洗浄ライン。自動化される前は皆さんの手作業によって支えられていたそうな、こうしたスタッフの日々の地道な作業が生命科学の発展を基礎から支えているのです。
人とマウスは同じ哺乳動物で、体を構成するタンパク質の遺伝情報も9割以上が共通しています。遺伝子の異常によって起こる病気の仕組みや症状にも多くの共通点が見出されています。人ではゲノム配列の解読が進み、一人一人の遺伝子配列の特徴と健康や病気の関係が明らかになりつつあります。一方、マウスの遺伝子にゲノム編集や遺伝子組み換え技術によって患者さんの遺伝子異常を組み込むことができるようになりました。こうした技術の進歩により、私たちは人の病気を再現するモデルマウスを研究者に提供して、病気の仕組みや治療薬の開発に貢献をしています。さらに遺伝子解明したモデルマウスは個々の遺伝子の働きを詳しく研究するのに大いに役立っています。
マウスは哺乳類を代表する実験動物として100年以上の歴史を持っています。私たちはマウスを通して将来の生命科学研究の基盤となり、人類の健康増進や病の克服に貢献したいと考えています。